Calvin cycle. Okay? And the Calvin cycle is the energy investment phase of photosynthesis. Okay? We talked about the light reactions yesterday. We said the light reactions were the energy capturing phase. This is the energy investment phase. Of photosynthesis. So we're going to take some high energy molecules like ATP and NADPH. So ATP and NADPH that were created in the light reactions. Are used to create carbohydrates. Sometimes the Calvin cycle is referred to as the dark reactions. Sometimes it is referred to as the light independent reactions. I think both of those are very stupid names because dark reactions is very wrong because it probably isn't going to happen very much at night. And light independent reactions makes a little bit more sense, except for the fact that it's, even though none of the reactions that are actually happening in the Calvin cycle require light, it still requires the light reactions to go on because you can't do it if you don't have NADPH and ATP, and you don't get those without the light reactions. So it just makes more sense to call it the Calvin cycle. And it's a cycle, and so you get that idea. Okay? All right. So I'm going to try something a little bit different right now, and that I haven't done yet today. And if it works, great. If it doesn't work, I'm sorry. So we're going to divide it up into 3 very easy steps. Okay? So the first step is going to be the carbon fixation step. So step 1, carbon fixation. Okay, in this step, you are going to take 3 carbon dioxides. Okay, that's what you need. 3 CO2s. All right? Those 3 CO2s are going to get added to a molecule that's called RUBP. Okay, so we're going to need 3 RUBPs. All right? Together, these 2 things are going to output through a series of reactions where you combine the carbon dioxide with the RUBPs. They're going to output some molecules that are called PGA. Okay? So, in the second step then, which is your energy investment step, you're going to add in some of those ATPs and some of those NADPHs. So we're going to add in 6 ATP and 6 NADPH. Okay? And then also those molecules that were created in the carbon fixation cycle, which were called PGA. We're going to need 6 of them. Okay, so these 2 things combined here create those 3 PGA or 6 PGAs. And then the last step, you are going to regeneration. And you're going to have these 6 PGAs, when they combine with ATP and NADPH, they're going to form something that is called G3P. Okay? And so you're going to end up with 1 G3P that is a product. You're going to end up with 5 G3Ps that are going to be used to regenerate, and you're going to need 3 ATPs to do that. And when you combine these 5 G3Ps with the 3 ATPs, those are going to create REBPs. Okay? So this is our overview here. This is all the stuff that you really need to know that's important. That your 3 CO2s and your 3 REBPs are going to create 6 PGAs. Then you're going to get 6 ATPs and 6 NADPHs to add to those. And that's going to create your 6 G3Ps. One of them is going to be a product, and 5 of them are going to be used to regenerate. Okay, so this is the overview. Now we're going to go into the nitty-gritty on like how we get this, and this will probably make more sense afterwards. Okay, step 1, carbon fixation. So these are, this is photosynthesis, so these are going to be autotrophs. So we know that the carbon is going to come from CO2 in the atmosphere. So we can say atmospheric CO2 is fixed, which means captured, by an enzyme called ribulose bisphosphate carboxylase. Okay, ribulose bisphosphate carboxylase is an enzyme, and it's almost always abbreviated RuBisCO, like that. Now, RuBisCO is a lot easier to remember than ribulose bisphosphate carboxylase, but it's not nearly as helpful, because if we look at the name ribulose bisphosphate carboxylase, it tells us exactly what's happening. First of all, we see that it's an ACE. Okay, because it's an ACE, we know that it's an enzyme. Then we see it says carboxylase. Okay, that means that it's an enzyme that's going to add a carboxyl group onto another molecule. Okay, and so then we just have to ask the question, all right, what molecule is that carboxyl group getting added onto then? Well, it's getting added onto the molecule ribulose bisphosphate. So ribulose bisphosphate carboxylase is the enzyme that adds a carboxyl group onto ribulose bisphosphate. Right? Carboxyl group is a CO2 group. Yep. All right. So, ribulose bisphosphate which is almost always abbreviated RUBP, is a 5-carbon chain. Okay? It gets a CO2 group added onto it. How many carbons is it now? It's a 6-carbon chain. So adding a CO2 molecule makes a short-lived 6-carbon chain. Okay? That 6-carbon chain is going to break down. It's going to break down into 2 3-carbon molecules. So 2 3-carbon molecules called PG, or sometimes it's called 3-PGA, or sometimes it's just called PGA. Those all mean the same thing. It's just depending on the textbook that you read. Okay, this is phosphoglyceric acid. Okay, whenever you see 3 in front of something like that, like 3 dash, like 3 dash PGA, that means that there is an important part to that molecule, and they want to tell you where it is. So if it says 3 dash PGA, it's talking about the phosphate. That's the important part, and it's saying the phosphate is going to be on the 3rd carbon, right? So 3 dash PGAs is a molecule called glyceric acid that has a phosphate group bonded to the 3rd carbon. That's why they put that 3 there. Okay, it doesn't mean that there are 3 of them. It doesn't mean that there's 3 carbons total. It means that that's where the phosphate group is. Okay, there does happen to be 3 carbons, but That's not what it means. All right. So this process here is going to happen 6 times. Okay? Or 3 times, rather, to make 6 carbons. Okay, so we have 3 RUBP plus 3 CO2 yields 6 PGA. Because each one RUBP is going to take... oh, wait, you mean like, why did we pick that? Save that for later. We talked first about there being 1, so 1 and 1, that's this, and that process happens 3 times to create a total of 6, right? And hang on to the reason why for a second. Okay? Step 2. This is our energy investment phase. In the energy investment phase, we are going to take those 6 PGAs and we are going to first phosphorylate them. So we can say that PGA molecules are phosphorylated by ATP to form a molecule called BPG. Okay, now that it's not really super important that you understand that this is an intermediate molecule here, okay, because it's not really going to come up in any test questions or anything like that. But it is kind of neat to know that, because later on when we do cellular respiration, we'll see that cellular respiration is just the exact opposite process. Also, it's pretty easy to learn this because you start out with this molecule called PG, or sometimes called PGA. This is phosphoglyceric acid. And then we add another phosphate to it, and all it changes is the B in front means bis. So sometimes in organic chemistry they use the prefix bis instead of di, okay, which just means 2. So this is bis phosphoglyceric acid then. So they just added another phosphate group onto it. Sometimes it's called 1,3-BPG. And in that case, they would just be saying, okay, there's a phosphate group on the 1 and a phosphate group on the 3rd carbon. This is just random stuff. You don't necessarily need to memorize any of that, but in case you're curious or you see it in the book, that's what it means. Okay? The second thing that happens is this molecule BPG molecules are reduced by high energy electrons in NADPH. Okay, we said NADPH is holding 2 high energy electrons. It dumps them onto BPG, and this creates a molecule that is called G3P. To form G3P, glyceraldehyde 3-phosphate. Okay? There are 6 PGAs that we started with, right? So we're going to have 6 PGA plus 6 ATP plus 6 NADPH yields 6 G3P. Of the 6 G3P that get created, 1 G3P is a product, meaning it gets to leave the cycle. 5 G3Ps are used in the next step. Okay? Step 3 is, what did I call it? Regeneration. Regeneration of RUBP. Okay, so the first thing that happens here is your 5 G3Ps, they combine with 3 ATPs. So 5 G3P are phosphorylated by 3 ATP in a series of reactions beyond the scope of this course. You do not need to know these by any means. You don't even need to know anything about them. Okay? If you're curious, you can look them up on Wikipedia. It's horrible. The total amount of reactions that take place in step 1 here and step 2 are about 20%, maybe 25% of the reactions that take place in the overall Calvin cycle, and the other 75% of the reactions in the Calvin cycle are the regeneration of RUBP. Okay? So you don't need to know them. Don't worry about it. It's no big deal. Okay? You do need to know that this will result in the production of 3 RUBPs. Then go on, start the next cycle. Okay, so we started with 3 RUBPs, we end with 3 RUBPs. Okay, we started with CO2s, 3 CO2s, and we end with our only product, which is this. Whoa, craziness. That's the only product of this whole thing, is 1 G3P molecule. Alright, so we got an animation. Let's watch the animation real quick. Step 1, here's Rubisco, here's the enzyme, right? Here is the CO2 molecules, here are the CO2 molecules, 3 of them. Right? Here's 3 RUBPs. The RUBPs are going to attach to this side of the enzyme. The CO2 is going to attach to this side of the enzyme. And Rubisco, which is ribulose bisphosphate carboxylase, will then join the CO2 with the ribulose bisphosphate. And that'll create a 6-carbon chain, which quickly breaks down then into PGA. 6-carbon chain breaks down into PGA. Like so. This happens 3 times. This leaves us with 6 molecules of PGA, phosphoglyceric acid. The next thing is we're going to add a phosphate group onto each phosphoglyceric acid to make BPG, right? And that's going to just be just like this, except for it's going to have a second phosphate on it. And then that's going to get reduced. And when it gets reduced by high energy electrons, that's going to add a lot of potential energy to the molecule, and it's going to create a very high energy molecule that's called G3P. Okay? This is going to happen 6 times. So ATP adds a phosphate, and NADPH reduces it, creates G3P. There's our 5, and now 1 is a product. Okay, the reason why 5 cannot be products is because if you count the carbons here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 carbons in these 5 G3Ps. Those were the 15 carbons that were in the ribulose bisphosphate to start with, right? 5 times 3 is 15. And so those carbons can't leave the cycle because they started the cycle. The only carbons that get to leave the cycle are the ones that came in from the outside world, which were these guys over here, CO2, right? So 1 carbon, 2 carbons, 3 carbons in CO2 corresponds to the 3 carbons that are in the G3P that get to leave. right? Make sense? So these then go through a series of reactions that you don't have to know about. They get combined with 3 ATP. And that produces our ribulose bisphosphate that can go on to start the next group. Okay? Now, if one G3P is a product, let's do our total products of Calvin cycle. One G3P. Now, it's also important to note that we have some other products here. What are the other products that we didn't talk about? Yeah.
I saw ADP on there.
ADP, yeah. So we used 9 ATP, so that means we're going to make 9 ADP. And what's the used up form of NADPH?
Wait, why ADP?
Because ATP turns into ADP when it gets used up. In 6 of them got used in the energy investment phase, and then 3 of them in RUBP. Okay, so this is going to become 6 NADP pluses as well. Okay, so these are the uncharged versions here. These are low energy molecules that need to get sent back to the light reactions. Right, so this gets sent back to light reactions.
Just the ADP and ADP?
Yep. And then this G3P thing, that's a 3-carbon chain. So we didn't talk about any products of 3-carbon chains. But if you have 2 G3Ps, G3P plus G3P, that's going to yield a glucose phosphate. Okay? And the cool thing about glucose phosphate is that glucose phosphate can become a lot of things. Because it has a phosphate group on it, we say that glucose phosphate provides its own energy source for dehydration reactions. So glucose phosphate provides the energy for dehydration reactions, meaning that it can form any glucose polymer without having to have an outside energy source. So they can form starch or cellulose, or it can just have that phosphate group cleaved off and it can become glucose or plain glucose. Okay, plain glucose was the thing that we said was created in photosynthesis, right? And so how many times do you have to go through the Calvin cycle to get 2 G3Ps? Twice, right? So that means that we're going to need 6 carbon dioxides total, 6 CO2s. Okay? How many NADPHs do we need? 6 per cycle. So that's 12. How many waters does it take to make 12 NADPHs? 12. We said it was going to make a glucose. When those 12 waters split, they form a bunch of oxygens, right? How many oxygens? How many O2s get made by that? 6 O2s. And this is almost the equation that you guys have seen, right? But it's close. We got to have more H2Os over here. There has to be 6 more H2Os over here. So where do those 6 H2Os come from? Well, when you regenerate the RUBPs, you have to go through a dehydration reaction in order to make those things combine together, which is going to create 3 waters per cycle, which creates 6 waters, right? And so then if you just take these waters here and you remove them from those waters there, you get 6 CO2 plus 6 H2O yields C6H12O6 plus 6O2. That's photosynthesis, right? Yeah. That's the equation we're all familiar with. And now we've seen exactly where each part is used. We know where the carbon dioxide gets used up. We know where the water gets used up. Right? Anybody have any questions over that? Okay.